show. Okay, a food web shows the interaction between all the species in a habitat. Okay, so it's a predator-prey relationship. So initially, the term was food chain, like from phytoplankton to zooplankton, zooplankton to fish, fish to humans. However, we now know that the interactions between predator and prey are very, very complex, and one prey can have a few predator, so it's no longer just a chain, but more of a web. Okay, so we learn the predator-prey interactions in order to understand the biomass, the carbon and energy transfer from the producers to the consumers. Okay, well, from there we can also determine how productive a habitat is, okay, relative to other places. Okay, so understanding the food web is crucial to ecosystem modeling and prediction systems. So we will be able to better assess any impacts to the habitat. For example, if a habitat is diverse, lots of microorganisms, lots of organisms inhabiting the area, we know that if there is abrupt change, okay, there may be a few species that will be able to overcome the change. So the uh, biological component of the habitat can still proceed, even though the diversity will be drastically reduced. There will still be life. Okay, so from there, if we understand the food web, we will be able to better predict the uh, the the robustness of the ecosystem to withstand changes. So a food pyramid tells you shows that essentially if it is a producer, they will usually form the highest biomass in a environment because they need to support the higher trophic levels, the consumers, whether they are herbivores or then carnivores. So there will be a reduction in biomass and abundance as you go up the trophic level. So from here, from the food pyramid, essentially what that means is if the primary productivity in the habitat is very big, okay, if the primary production is very high, theoretically it will be able to support more trophic levels. Okay, there will be more diversity, more trophic levels, more diversity in that habitat. So we have covered this where we show the different primary producers in the oceans. Okay, we show that these are the main components of primary producers. We have the prokaryotic, we have the eukaryotic, and each of them have their own niche. Okay, low light, uh, end-limiting environments. Okay, with silicate, without silicate, we can have this different uh, predominant primary producer in different types of environments. Primary producers as always being in the euphotic zone where there is light. Okay, and we also know that it requires certain nutrients. Okay, and we also know that, so if I were to ask you what are the factors that can affect primary producers in the sea, you'll of course first have to uh, you can categorize your answers as whether they are prokaryotic primary producers or eukaryotic primary producers, whether they are Prochlorococcus, Shinnicoccus, diatoms or dinoflagellates. So because each of them will have different factors that can affect them. But generally, there are commonalities among all these components. The first is the requirement for light. Okay, they require light for photosynthesis. Therefore, they grow well, they thrive in the euphoric zone, where where the light is enough for photosynthesis, uh, for productivity, for primary production. Okay, so first factor is light. Of course, you also need nutrients, whether nitrogen, phosphorus. For silicate is of course only for mainly for diatoms. So this is the second factor, the availability of nutrients. Two, the availability of nutrients that can affect primary production in the seas. Some students will state that CO2 will also affect, but usually CO2 does not limit uh, primary productivity because CO2 is easily available. Okay, there are still no occasion where CO2 becomes limiting for primary producers. So we have one factor here, two factor here. What else do you think can affect? When you have light, the temperature is also important. Okay. That's why most of the primary production increases spring and summer, but is lower in winter and when the waters are colder. Okay, so that is one factor. Whether salinity affects, that is of course dependent upon the location of your study site. If you're working in an estuary, of course then salinity becomes important because the salinity in a river mouth will change regularly, and the change occurs over a wide range. It can easily move from 30 to 5 parts per thousand salinity. Okay, so there is a wide range there. So the changing of the salinity can affect primary production. But this is very site specific, usually for estuaries, river mouths. Okay, for open oceans, salinity does not really change. Okay, so it doesn't affect so much. Okay, one factor that is important is Grant's effect. So you must remember that phytoplankton are relatively larger than bacteria. Then in natural environments, phytoplankton will tend to sink. Okay, so naturally phytoplankton will sink. Bacteria will not. Okay, bacteria does not sink in the sea. Okay, because they are not heavy enough, they will not sink. Bacteria sinks when they are attached. When they are attached to a larger particle, they may sink. But phytoplankton by itself will sink. So what happens? When it sinks out of the euphoric zone, it will be away from the light, then that is detrimental to it. It cannot produce, it cannot carry out primary production, it cannot replicate. That's what Grant's effect means. Grant's effect is the observation that the phytoplankton has to be kept within the euphoric zone in order for it to maintain its primary production. Okay, that's why in in turbulent waters, in turbulent waters, where there's a lot of mixing, sometimes even though the nutrients are high, because of the mixing, the nutrients are high, but you do not get a lot of primary production here because the phytoplankton is not given enough time in the euphotic zone to replicate, to carry out primary production. Okay, even before it is able to replicate, it is being brought down away from the euphoric zone. So you do not see any significant increase in primary production. So Grant's effect is one of the is an important factor that influences primary production and primary producers in the sea. Okay, the ability, the observation that primary producers have to be kept in the euphoric zone long enough for them to replicate. Okay, to carry out primary production. So this is called the Grant's effect. So of course, these are physical chemical variables that affect them. Physical chemical variables. Okay. You also have, so this can be considered abiotic factors. Okay. You of course also have biotic factors. So when in terms of biotic factors, usually it is the microzooplankton, flagellated, that eats the diatoms, so grazes. The main ones are microzooplankton. You can have zooplankton, copypods. Copypods will take up zooplankton. Okay, so microzooplankton takes up, consumes the primary producers. Okay, so they form a part of the biotic factor that can affect primary production. Other than grazers, you also have your virus. Viruses that infect phytoplankton. Okay. Viruses are termed important, especially after a bloom. Okay. So when there is a phytoplankton bloom, when the phytoplankton numbers increase significantly, viruses are known to terminate the bloom. Okay. So when you have a bloom event, viruses are known to terminate it, to be able to stop the continuation of the bloom event. That's where the importance of viruses as a biotic factor controlling primary production. So the classical food chain, from the understanding of ecology, and it was still published in 1972 in biological textbooks. Okay, where you have the phytoplankton will be eaten up by the microzooplankton, then the microzooplankton will be eaten up by copypods, and the copypods will be eaten up by the fish, and the fish will be eaten by man. Okay, so this is a very, very simple, linear, classical food chain. Okay. So the transfer of energy and biomass in the classical food chain is very straightforward. Okay, where you can see the biomass here, eaten up here, and eaten up here, here, and all the way here. Of course, you realize that you have to understand that the energy transfer is not efficient. you lose carbon, you lose energy at each transfer. That's why we have to eat more of fish in order to sustain ourselves, because you lose carbon, you lose energy through heat and your respiration. You respire CO2 when you metabolize. So you lose the carbon, you lose heat, energy in the form of heat with each transfer from each trophic level. That's why uh, that's why the primary producers have to be large in order to support the subsequent trophic levels. Okay, that's why if the primary producers are a small component, then that habitat is not productive. Okay, and will not be able to sustain many types of organisms. However, in a classical food chain, the role of bacteria. Okay, so we're talking about our understanding in the past. The role of bacteria is very straightforward. We know all these components will defecate. There is also that there are fecal pellets from copy pots from the fish, from and also when they graze. Okay, if you look at the size difference, phytoplankton are large, microzooplankton are small. So when they consume, when they graze on the diatoms or on the phytoplankton, there's a term for the feeding mechanism. They call it sloppy feeding. So when they eat, they are very sloppy. There are pieces of diatoms that will uh, be left be left be discarded into the sea. So it is not efficient. It, it's not as if the microzooplankton engulf the whole phytoplankton. No, when they bite pieces of the cytoplasm, the cytoplasm of the diatom, the phytoplankton, the pieces of cell wall, pieces of tissue will be discarded. So that's called sloppy feeding. All this will contribute to the detritus, the particulate organic matter in the sea. And in the past, the role of the bacteria is essentially to uh, degrade this, utilize this detritus, and then recycle all the nitrogen and phosphorus back to the phytoplankton. So that was the role of bacteria in the uh, classical food chain. Okay. Okay. So, but then certain observations made the classical food chain untenable. Okay, you cannot unacceptable. Okay, certain things don't match, don't fit. First of all, observations like the smaller size phytoplankton are important. You see more of them in the seas. Then when they carry out respiration experiments where they put samples of water, they size fractionate it, which means they use filters and separate the water, and then they observe the respiration. And they found out that the the smaller fractions are actually very important. There seems to be more respiration in the less than 300 micron fraction. That means we are missing something. That means because all these are larger than the 300 micron. So we are missing the smaller fraction that seems to respire more. So something is not right here. Okay, and also that we begin to realize that in the waters there is a very high amount of DOM and POM pool. Okay, so a lot more organic matter than previously known. Okay, and the abundance of bacteria. So in the past, if you do culture, you will only get 10 power 3 cells per mil of bacteria. But when you do direct count, even under hemocytometer, you realize the number of bacteria is easily power 5 to power 6 cells per mil. So definitely bacteria is more important than previously thought. Okay. So these observations were uh, compiled and put forward by Pomeroy in 1974 as a paradigm shifting paper where he proposed that the role of microbes are more important than previously thought. Okay, so he was saying that the classical food chain is not tenable anymore. Okay, something, and he proposed a new one where the the particulate organic matter and the dissolved organic matter are utilized by bacteria, and then this bacteria is brought back into the zooplankton via the microbial loop. Okay, the microbial loop is a term coined by Farooq Azam in 1983. Okay, so the first there was recognition that the dissolved organic matter pool is large. Okay, and the contributors to the dissolved organic pool includes phytoplankton. It seems that when phytoplankton is carrying out primary production, up to 50% of its primary production is actually leached. Okay, can be leached out from its body. Okay, so that contributes a significant portion of the labile fraction of DOM. And from sloppy feeding and also from discharge, you also have contribution to the DOM pool from microzooplankton and also from zooplankton. So if bacteria only degrades detritus, then this organic matter pool is wasted. Okay, no other organisms, no other component is able to utilize them. Okay, so it is bacteria that is the one that utilizes this DOM, okay, repackage it as bacterial biomass, and then this bacteria is grazed by protozoa, okay, smaller protozoa, and this protozoa is then eaten up by microzooplankton, by the ciliates especially. So when at this stage, the carbon that is here is being transferred back into the food chain. Okay, so the role of bacteria became more important. So the role of the microbes is being accounted for. Okay, we are beginning to understand why this fraction, the microbial fraction, respires more because they're very active. Okay, they degrade, they take up the DOM, repackage it as bacterial body biomass, and then it's eaten up by protozoa and then transferred back to microzooplankton. So this is the microbial loop, the term microbial loop, where DOM that is essentially lost in the sea, being reused, repackaged by bacteria back so that the carbon and energy here is transferred back to the food chain. Okay, that's why the role of bacteria is very important. Okay, yeah, bacteria is important, and more studies need to be carried out in order to understand their function in different habitats. Okay, so it's important in a sense there is regeneration of energy and biomass from an otherwise unutilizable source. Okay, that's important here because without bacteria, the whole DOM pool is like that. It will just be there. No other component uses it. Okay, so bacteria is an essential link in the microbial food web in the seas. Okay, but this is also not only limited to seas because all aquatic habitats also have the DOM pool and also have the microbial food web. But most of our understanding initially was from uh, studies carried out in the sea. Okay, so Pomeroy's paper is a landmark paper uh, where the role of microbes, where the important role of microbes in the seas was being proposed. Uh, Azam, important for the coining of the term microbial loop, okay, that shows the important role of bacteria to repackage DOM back to the classical food chain. Okay, the microbial food web doesn't end here. Okay, we still have to account for virus. Because viruses are the most abundant component in the seas. Okay, so although there are viruses for each of these component, we are more concerned with the viruses the phages, okay, bacterial phages that infect bacteria, okay, and lyse it, okay, through the lytic cycle, and then when it lyse the bacteria, the cytoplasm of the bacteria is being recycled back to the DOM pool. So this becomes another loop, okay, another loop. And this is termed the viral loop. Because now that we have determined bacteria as important, we need to understand also the role of viruses because we need to understand the role of the viruses because viruses will affect bacteria. So we know bacteriophages can undergo either lytic or lysogenic cycle. Okay, so lysogeny decrease with temperature and decrease with eutrophication. In tropical countries where the waters are warmer, lysogeny plays a less important role as compared to lytic cycles. Okay, and in 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 places that have high eutrophication, for example, near shore waters with higher amount of nutrients, the lytic cycle is also more important as opposed to lysogeny. Okay, so viruses are important because they can contribute up to 50% of bacterial mortality. So this will actually accelerate the turnover of the organic matter pool via the DOM loop or the viral loop, where the DOM goes to bacteria, the bacteria goes to virus, and the virus lies and released to DOM again. So now we know that some of the phytoplankton are also mixotrophs. Some of the microzooplankton, the protozoas are mixotrophs, which means they are capable of both phototrophy and heterotrophy. It just further complicates our understanding of the marine microbial food web. However, these are still minor components. Okay, generally the story is still the same. Okay, so this is the uh, marine microbial food web as it is as per our understanding now.